Click for IB Biology. This is our third video in the eukaryotic cell structures, looking at organelles. In this one we're going to be looking at plant structures such as the vacuole, the tonoplast, which is the membrane around that vacuole. We'll also take a look at the cell wall, although theoretically the cell wall is not considered to be an organelle as it's actually outside of the cell membrane. It's an extracellular structure, so not strictly speaking an organelle, but one that we should be aware of. And then these curious structures called Plasmodesmata, which exist in plant cells. There's a blank PDF of this presentation down in the description that you can annotate with your own notes as we go through this. But then there's also a quiz, 10 questions with answers that you can use to check out your understanding. So let's take a look at this structure called the vacuole. The plant vacuole occupies a large region of the cell volume, and you can see here in this image there's actually two cells shown in my drawing here. The plasma membrane would be running approximately in this region here like so, and a cell wall either side. I think we can basically make that out. But the actual vacuole for the first cell is here, and my pen line is now drawing around each of the vacuoles. I'm actually just going to stop this one because this structure here is the second item on our list, which is the tonoplast. Now the tonoplast is a cell membrane structure with exactly the same features and characteristics as the plasma membrane or cell membrane. And the tonoplast acts as a way of regulating what goes into and out of the vacuole. The vacuole should be thought of as a storage area, and typically you would store things like sugars, amino acids, and perhaps more complex molecules within this particular space. So the regulation of the content is through the vacuole. The water content is a feature of the exchange of water due to osmosis, but the vacuole occupies a large part of the storage region of the plant cell structure. As mentioned, the cell wall is not thought of as an organelle. It's a cellulose structure. It's an extracellular matrix, if you like. It's secreted outside of the plasma membrane is here. So this is on the outside. It's a network of cellulose fibers which interconnect, providing a very rigid structure, and indeed is of tremendous importance in providing support to plant cells. And we'll come across cellular cell walls when we consider the processes of osmosis. In the image here, we can see that the chemical nature of cellular cell walls with the electron microscopy leaves them with very little contrast. It's the clear region here, always recognized by its width. There's usually a clear area between two cells, well, a fairly large distance between them, which is the cellulose cell wall. But if one looks carefully, there are these, I'll not draw over the top of them, I'll just pinpoint their location. These structures here, and these are so-called Plasmodesmata. These are cytoplasm extensions from this cell to this cell. So the cytoplasm of plant cells is actually connected, which will be a new concept to many students. The Plasmodesmata connection has a diameter of about 20 to 40 nanometers. This allows ions, charged particles to move between them, sugars and amino acids, and so therefore nutrient can be regulated and exchanged through the plasmodesmata. And it's also a channel for the movement of water, which we will feature later in our videos on osmosis. So that will be almost certainly a new feature for students, the idea that the cytoplasm of two cells is connected. Notice that the size of those does not allow for the movement of organelles between the cells, but molecules, yes. So that takes us to the end of our look at eukaryotic organelles and trying to recognize them within the cytoplasm. Links to the next video, to the channel, and to the playlist are featuring here for you to choose your next study.